So let us start with our discussion. So what we are going to start with today is basically the prokaryotic genome organization. As I told you that my way of dealing with this topic is going to be wherein we first discuss about the prokaryotic genome, and then we will go to the eukaryotic genome, and finally we will talk about the DNA replication process. So that's going to be the order in which I will deal with this topic with you guys. Okay, so to begin with, we have to know or understand what basically prokaryotes are. We know that prokaryotes basically are those organisms which do not have a well-defined nucleus, right? They do not have a specialized set of compartments like we talk about in case of eukaryotic cells. We speak about presence of endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes. These are all not present in the prokaryotes. They do not have membrane-bound organelles, which we normally find in eukaryotic cells. Their genome is lying free in the cytoplasm in the form of a nucleoid, right? Now, under prokaryotes, we have the bacteria, the modern bacteria, which we call as eubacteria, right? And they basically are characterized or classified into two sections. We call them as gram-positive organisms, gram-negative organisms. And we also know that blue-green algae, which we popularly call as cyanobacteria, have also been included under prokaryotes itself. But along with the eubacteria, we have Archaebacteria, which are primitive types of prokaryotic organisms, which probably have been the first set of organisms to have evolved on Earth. Okay, and they are not comparatively, explored as compared to what we have done with the modern day bacteria. We know so much about eubacteria, but there is not much work that has been done on Archaebacteria because first, it is not very easy to cultivate them. I don't say it's impossible, but they have specific requirements for their cultivation in terms of conditions, which may be difficult to mimic because most of them are extremophiles. Now, what do you mean by extremophiles? They grow in extreme conditions like extreme acidity, extreme alkalinity, extremely high temperature or very low temperature, or they require a lot of salt concentration, okay, or they require a lot of pressure. Okay, we call them as barophiles. So the conditions that they may need, it may be very difficult to mimic it in the lab, because of which cultivating Archaebacteria has been a challenge. But that doesn't mean that scientists have not been able to cultivate it at all. There are a lot of people who have also cultivated Archaebacteria in the lab successfully. If you want to find Archaebacteria, the easiest sites would be hot water springs or acidic springs. You can also find a lot of Archaebacteria very deep in the ocean, near the ocean floor, where there's a lot of pressure. You can also find a lot of Archaebacteria in the glaciers, where the temperature is very, very low. Okay, so we need to understand that Archaebacteria are still prevalent. Don't think that they are not around us. Rather, I would say that they are also inside us. Many people have started looking for archaebacterial species which live in conditions like our gut, our intestine. So there are several research groups, especially with the advent of microbiome studies. I'm sure you all know what is a microbiome, right? Microbiome is a community of microorganisms that is prevalent in specific part. So human microbiome is being studied to a great extent. What is our skin microbiome? What is our gut microbiome? All this is being analyzed, and we are trying to link the data that we are getting from there to different kind of diseases, disorders that are prevalent amongst human beings. Okay, so with the advent of human gut microbiome studies, people have found a lot of kinds of Archaebacteria also, which are going to persist in our gut. Okay, so it's not that you have to look out for external samples. Many a times you yourself might be a sample for Archaebacteria. Okay, moving ahead. Now, if you think about the eukaryotic genome and the prokaryotic genome, they are not one and the same. Okay, there are some similarities, but there are several differences as well. especially in terms of how the prokaryotic genome is organized, how it is packaged. There is a lot of difference when it comes to the packaging of the eukaryotic genome as compared to that of the prokaryotic genome. Okay, now no doubt that we use this word chromosome so often to describe the DNA packaged along with the proteins, because we know that DNA is never alone. There are some proteins always associated to it. So this complex of DNA and protein structures is what people normally refer to as a chromosome. But let me tell you that this is a misnomer, because prokaryotic chromosomes are not very similar to that of eukaryotic chromosomes. There are a lot of differences. So it should not be interchangeably used is what we are trying to say over here, because the typical prokaryotic genome is a single circular DNA molecule. Okay, and it's not enclosed in a compartment, it is not enclosed in a nucleus, it is localized within a structure that we call as nucleoid. No doubt that here also we have proteins which are enclosing this DNA and packaging it in that circular form, which we refer to as nucleoid. But let me tell you that there are a lot of differences in the kind of proteins that are associated with the DNA in case of a prokaryotic cell. Okay. Now, what we need to understand is that whatever data we have about the nucleoid, whatever data we have about the prokaryotic genome, that comes from E. coli. I always tell students that unfortunately, whenever it comes to bacteriology, we end up learning so much about Escherichia coli that we should call it as Escherichiology rather than calling it as bacteriology. Because you see, maximum of your experiments, they run around E. coli only. So E. coli has become the model organism, a representative when it comes to the bacterial species. But that doesn't mean that we do not have, any other organisms which can be used as a representative. Many a times you may also find some differences amongst the bacterial, organisms itself. So we surely have data for E. coli, which is a general data which represents for the bacteria, but that doesn't mean it's going to be the same in all kinds of bacteria. So you need to study more and more types of prokaryotic genomes, which will remove this misconception that is there amongst the people that हाँ E. coli का genome पढ़ लिया मतलब सारे बैक्टीरिया का genome same होगा, ऐसा होता नहीं है. Okay, we need to understand that bacteria to bacteria there are going to be variations. Yes, the general organizational features may remain the same. Okay, but there are going to be differences, few differences here and there when you compare to prokaryotic genome itself. Okay, now this is basically a transmission electron micrograph that you can see, which shows you the cross section of a dividing E. coli cell. Okay, and you can see that the lightly stained region is basically nothing but the nucleoid. Okay, that's indicating the presence of nucleoid in the E. coli cell. Now, a prokaryotic genome, a prokaryotic cell by itself is so tiny as compared to a eukaryotic cell. You know that the prokaryotic cell is going to be very, very small in size, right? So, under these circumstances, I already have tiny space to accommodate the genome. Okay, so genome को अगर मुझे इस छोटे से जगह पर squeeze in करके fit करना है, तो उसका packaging compact होना बहुत जरूरी है. It is going to be very, very important to squeeze in the genome, because they have shown you the example that E. coli का जो cell है, उसकी dimension is 1 into 2 micrometers. But if you see the circular E. coli chromosome, it has a circumference of 1.6 millimeters. Cell का size micrometer में और circular E. coli chromosome का circumference millimeter में. तो अगर मुझे इतने बड़े मॉलिक्यूल को छोटे से सेल में ठूसना है, उसको पैकेज करना है, तो मुझे जितना कॉम्पैक्ट पैकेजिंग हो सके उतना कॉम्पैक्ट पैकेजिंग करना पड़ेगा. यूकैरियोट्स का भी सिनेरियो यही है. यूकैरियोट्स में भी सेल बहुत छोटा है और जो जीनोम मुझे उसके अंदर पैकेज करना है वो बहुत ही ज्यादा बड़ा है. तो उसको कॉम्पैक्ट पैकेजिंग करने के लिए वहां पर बहुत सारे डीएनए बाइंडिंग प्रोटीन्स रोल प्ले करते हैं. ओके, और हम उसको न्यूक्लियोसोम नाम के एक स्ट्रक्चर में कॉम्पैक्टली अरेंज करके सेल के अंदर फूस देते हैं. So we need to understand that this logic has to apply with the nucleoid also. I have to package the DNA in such a way that they are compact so that they can remain in that tiny cell. Now, the first thing that was recognized with the E. coli genome was that the genome is supercoiled. Now, what is supercoiling? Supercoiling is wherein either you are introducing more twists and turns into the DNA, or you are releasing few turns in the DNA, either of it. So if you are adding turns to it, if you are adding turns into the DNA double helix, you call it as positive supercoiling. So that becomes more compact. And if you're removing the turns, if you're releasing the turns, then you call it as negative supercoiling. So positive supercoiling is when you introduce the turns. Negative supercoiling is going to be when you remove the turns. Now, please understand, whenever you're doing supercoiling, if you're adding the turns or if you're removing the turns, it is going to bring about torsion. क्योंकि देखो ये टर्न मैं कहीं से भी रिलीज कर सकता हूँ बीच में, और कहीं पे भी बीच में टर्न ऐड कर सकता हूँ, तो बाकी के मेरे डीएनए मॉलिक्यूल पर स्ट्रेस आने का चांसेस है, टॉर्शनल स्ट्रेस आने का चांसेस है, क्योंकि मैंने ओवर वाइंडिंग या अंडर वाइंडिंग किया है एक पर्टिकुलर जोन में डीएनए में, तो डीएनए का बाकी हिस्सा इज गोइंग टू फील दैट स्ट्रेस. सो अगर एक लीनियर डीएनए स्ट्रक्चर होगा, जैसे कि यूकैरियोट्स में होता है, हमारा डीएनए लीनियर है, सर्कुलर नहीं है, तो मैंने कहीं पे भी अगर सुपर कॉइलिंग इंट्रोड्यूस किया, आइदर मैंने ज्यादा टर्न्स इंट्रोड्यूस किए या कम टर्न्स किए, तो जो भी बाकी हिस्सा है डीएनए का जो टॉर्शनल स्ट्रेस फेस कर रहा होगा, वो उस स्ट्रेस को रिलीज कर सकता है. कैसे रिलीज करेगा? लीनियर एंड्स पर, टर्मिनल एंड्स पर दे विल रिलीज द स्ट्रेस बाय अनवाइंडिंग और by underwinding it, okay, by introducing more turns or by releasing some turns. So तुमने अगर बीच में कई टर्न्स इंट्रोड्यूस किए हैं और बाकी पार्ट्स पर अगर स्ट्रेस आ रहा है, तो वो टर्मिनल एंड जो है लीनियर डीएनए मॉलिक्यूल का, वो उस स्ट्रेस को बैलेंस आउट कर सकता है by bringing about some underwinding or by winding up more, depending on whatever the condition is. बट अगर स्ट्रक्चर सर्कुलर है, अगर उसके पास टर्मिनल एंड्स ही नहीं है, तो फिर इस स्ट्रेस को कैसे हैंडल करें? आर यू अंडरस्टैंडिंग द प्रॉब्लम ओवर हियर स्टूडेंट्स? सबको समझ में आ रहा है ये कॉन्सेप्ट कि हम यहाँ क्या डिस्कस करने की कोशिश कर रहे हैं? यस और नो? Others, only 2 people. Rest, what are you doing? See, a response for me is important, okay? Otherwise, we'll stop then and there. Otherwise, it's useless carrying the discussion ahead. Why am I putting so much efforts then if you can't respond? Anyways. So, you know, for a circular molecule, as it's going to be difficult to release the torsional stress, what will it do? It needs to have some weight. So the circular molecule will wind around itself. So अगर मेरे पास एक सर्कल होगा, वो खुद को घुमा लेगा, वो सर्कल ही घूम जाएगा. लीनियर डीएनए में क्या हो रहा है? टर्मिनल एंड्स पे अनवाइंडिंग हो रहा है या वाइंडिंग हो रहा है टू रिलीज दैट स्ट्रेस. सर्कुलर मॉलिक्यूल के पास कोई टर्मिनल एंड है ही नहीं, तो वो क्या करेगा? वो सर्कल को ही घुमा के ट्विस्ट कर देगा, सर्कल में वाइंडिंग चालू हो जाएगा. ओके, आई विल शो यू द इमेज टू मेक यू अंडरस्टैंड दिस. दिस इज योर सर्कुलर डीएनए. ओके, ये तुम्हारा सर्कुलर डीएनए है, और वो सर्कुलर डीएनए में हमने स्ट्रेस इंट्रोड्यूस किया एट वन पॉइंट बाय अनवाइंडिंग दिस रीजन. ये रीजन को हमने अनवाइंड किया, तो बाकी बेचारे जोन पर स्ट्रेस आने लगा है अनवाइंडिंग की वजह से. तो ये स्ट्रेस को कैसे रिलीज कर रहा है? सर्कल में ट्विस्टिंग ला रहा है. यू कैन सी व्हाट इट इज डूइंग. तो सर्कल में ट्विस्टिंग लेकर आ रहा है, so that the stress that was created by this few turns of unwinding can be released. Okay, so supercoiling is indeed an ideal way जिससे हम मॉलिक्यूल्स को कॉम्पैक्ट बना सकते हैं, क्योंकि एक सर्कल जितना जगह ऑक्यूपाई करेगा, ये सुपरकॉइल्ड फॉर्म उतना कम जगह ऑक्यूपाई करेगा. तो तुम डीएनए को उतना कॉम्पैक्ट करते जाओगे, ओके, बाय इंट्रोड्यूसिंग सुपर कॉइलिंग। तो सुपर कॉइलिंग जो है ये सबसे पहले इकोलाई में प्रेजेंट है, ये 1970s में प्रूव हुआ था, okay, and they had isolated nucleoids from E. coli and they had checked the supercoiling that is present, and subsequently it was confirmed that inside the cell also it is present in that supercoiled format only after 11 years, क्योंकि पहले आइसोलेटेड न्यूक्लियोइड में उन्होंने वो सुपरकॉइलिंग देखा था, यानी कि जो न्यूक्लियोइड सेल में से बाहर निकाला, but they wanted to check that if it is present in the same format inside the cells also, inside the living cell also, तो वो प्रूव करने के लिए उनको 11 साल लगे, 11 years. So in E. coli, the supercoiling is brought about by an enzyme, okay, and that enzyme basically is DNA topoisomerase, which is also called as DNA gyrase. अब इस एंजाइम के बारे में हम रेप्लिकेशन के दौरान और डिटेल में पढ़ेंगे, so right now we will not get into it, okay, because we will be speaking about this enzyme in more detail when we speak about DNA replication. But keep in mind that this supercoiling, whether it is winding or unwinding, that is basically generated and controlled by this particular enzyme, which we call as DNA topoisomerase, or we also call it as DNA gyrase. Okay? Now, please understand that supercoiling can continue till the point of time your DNA is intact. If your DNA is not intact, if there is a break in the DNA that is introduced, then they do not have freedom to carry out supercoiling to a great extent. They can carry out supercoiling effectively till the time DNA is intact. If there is a break in the DNA, supercoiling will start getting affected. क्योंकि ब्रेक मतलब क्या, कहीं पे अगर वो टूट चुका है तो ऑटोमेटिकली जो तुमने वाइंडिंग किया हुआ है वो वहीं पे लूज पड़ने लगेगा, सुपरकॉइलिंग का मतलब खत्म हो जाएगा, ओके। तो जितने ब्रेक्स होंगे उतना तुम्हारा सुपरकॉइलिंग जो है वो रिड्यूस होते जाएगा, ओके। सो यू हैव टू अंडरस्टैंड दिस, दैट if there are breaks in the DNA, it is difficult for supercoiling to continue or to exist there. Now, bacterial DNA is attached to proteins, okay, and the proteins will not allow the DNA to relax or to come back to their loose forms. These proteins are responsible to keep them in that tight form, in that supercoiled form. But if there is a break in the DNA, then what happens is these proteins are not able to attach very firmly. The proteins detach, and as the proteins detach, you will see that there is loss in supercoiling. The compact structure becomes loose. So उसको intact उस form में hold करने के लिए proteins की जरूरत है, और proteins के साथ साथ DNA का intact होना जरूरी है. अगर DNA intact नहीं रहा, then it's all gone. So this can be tested using a particular chemical which is called as trimethylsorelane. Okay, trimethylsorelane, when it is activated by a wavelength of light, because it is a chemical which is photoactivated. So when you expose it to 360 nanometers, which is a UV light, you will see that trimethylsorelane goes and binds to double-stranded DNA very effectively. And it is going to bind to the DNA where there is supercoiling. तो यानी जितना trimethylsorelane DNA को bind होगा, उतना तुम्हारा वहां पर torsional stress है, उतना supercoiling है, ये वो बता रहे हैं. So तुम degree of supercoiling measure कर सकते हो by checking how much of trimethylsorelane goes and binds to your DNA. Because it can bind to the DNA only if there is supercoiling. If there is no supercoiling, it will not bind. So if there is a break in the DNA, supercoiling is reduced, you will see that the attachment of trimethylsorelane will also decrease. So trimethylsorelane has been used since a long period of time to check the degree of supercoiling that is present in the DNA. More the supercoiling is, more is going to be the binding of trimethylsorelane. Lower the supercoiling is, less the amount of trimethylsorelane will bind to. Okay, so even people have done that. If you use radiation to introduce breaks, single-stranded breaks into DNA molecule, you will see that the amount of trimethylsorelane that can bind to the DNA is going to be less. So more you increase the radiation, more less amount of trimethylsorelane can go and bind to the DNA. So it is inversely proportional to the radiation dose. जितना ज्यादा रेडिएशन होगा उतना कम ट्राइमिथाइलसोरलेन डीएनए को बाइंड कर पाएगा. क्यों? क्योंकि जितना रेडिएशन तुम एक्सपोज करोगे, सब्जेक्ट करोगे, उतने ब्रेक्स होने लगेंगे. जितने डीएनए में ब्रेक्स होने लगेंगे, सुपर कॉइलिंग उतना कम होते जाएगा. जितना सुपर कॉइलिंग कम होते जाएगा, उतना ट्राइमिथाइलसोरलेन का बाइंडिंग जो है वो भी कम होते जाएगा. Okay, so you need to understand this. Okay, and you can see here, this is a graphical representation. They have shown you the percentage of trimethylsorelane binding on y-axis, and they have shown you the radiation dose on the x-axis. So you can see as the radiation dose increases, you can see that the amount of trimethylsorelane that can bind to the DNA is also decreasing. 10 के बाद तो शून्य ही है, क्योंकि उतने ब्रेक्स इंट्रोड्यूस हो चुके हैं और सुपर कॉइलिंग बहुत ही कम हो चुका है. सुपर कॉइलिंग नहीं रहेगा तो ये केमिकल वहां जाकर बाइंड करने वाला नहीं है. So please understand that your nucleoid of E. coli, it is present in, the form of domains. Single domains है DNA में, nucleoid में. Okay, you have to understand that ये domains होने के वजह से it's like segregations. तो एक ब्रेक से पूरा डीएनए का सुपर कॉइलिंग नहीं अफेक्ट होता. कई ब्रेक आया तो उस रीजन का सुपर कॉइलिंग अफेक्ट होता. समझो अगर ये डोमेन्स नहीं होते, ये सेग्रीगेशंस नहीं होते विद इन द डीएनए, तो कहीं एक ब्रेक काफी होता पूरे डीएनए के सुपर कॉइलिंग को डिसॉल्व करने के लिए. मतलब कहीं एक टुकड़ा हुआ, पूरा डीएनए का सुपर कॉइलिंग खत्म. बट ऐसा है नहीं. डीएनए में भी डोमेन्स है ई कोलाई, जिसके वजह से एक जगह के ब्रेक से असर उसके इर्द गिर्द ही होगा, लोकलाइज्ड इफेक्ट होगा. पूरे डीएनए के सुपर कॉइलिंग पर तुरंत इफेक्ट नहीं होगा. हाँ, लेकिन अगर मल्टीपल ब्रेक्स है डोमेन में, देन ग्रेजुअली पूरा डीएनए का सुपर कॉइलिंग खत्म हो जाएगा. लेकिन एक ब्रेक से पूरे डीएनए का सुपर कॉइलिंग खत्म होने वाला नहीं है, बिकॉज द डीएनए इज नॉट अ सिंगल डोमेन, इट इज मल्टीपल डोमेन्स. So one break will affect that localized zone super coiling. Multiple breaks will start affecting several domains, and gradually the entire super coiling will reduce. Single domain होता था तो एक break से it would have been yes or no answer कि super coiling पूरा खत्म या super coiling है. अगर एक domain होता था, but एक domain का case नहीं. It has been divided into multiple domains. Okay, now. The current model of E. coli domain that we have, it's attached to a protein core. Okay, the protein core is at the center and the DNA is radiating out. Okay, I'll show you the image of how it looks. This is how it looks. So the pink colored thing that you can see at the center is the protein core, which holds the DNA fragments, and you can see the DNA radiating out. Okay, so these are all supercoiled DNA that you can see. But you can see one DNA section, it is loose. It's not coiled, it is completely loose. Because there might be a break there, because of which the supercoiling is lost. So ये सारे मेरे domains हैं, अलग-अलग domains हैं same organism के genome के. एक breakage के वजह से बाकी domains unaffected. They are not facing the brunt of it. Only the part where there is a break, you will see that the supercoiling is lost. The rest of the domains are intact. That is what I'm trying to say. Now, each loop is going to have around 10 to 100 kilo base pairs of supercoiled DNA. Okay, so that's the amount of DNA that you would get when you unwind it completely. On 10 to 100 kilo base pairs. That's the reason why it's not the same for every domain. That's the reason why you can see some domains are short, some domains are long. Because the amount of DNA is not going to be constant. It's going to change from one loop to the other. Okay. Now the protein component, the protein core of your nucleoid, is made up of nucleoid associated proteins. There are several different types of proteins which form that protein core, which is responsible for organizing the nucleoid in that specific fashion of a prokaryotic. Now the early findings that were done about this protein core, it was found that there are proteins from the HU family, okay, which is a family of proteins, and These proteins have a lot of amino acid similarity to the histone proteins. Histone proteins are DNA binding proteins which we see in eukaryotes, which are responsible for packaging your DNA to that nucleosome format. In prokaryotes, we do not have histones. We have histone-like proteins, but we don't have histones. The amino acid sequences may be similar to what it is in case of histone proteins, like histone H2B in eukaryotes, but it's not exactly histone protein. We have histone-like proteins that are present in case of prokaryotic protein code. You have to remember that. Okay. Now, the HU protein that may be present here, they normally form a dimer. What is a dimer? You have two subunits. Okay. So it is HU alpha or HU beta. Okay, there can be two HU alpha proteins together forming a dimer, or two HU beta proteins together forming a dimer, or you can have one HU alpha, one HU beta coming together to form a heterodimer also. अगर दोनों components alpha के तो तुम उसको homodimer बोलोगे, या दोनों बीटा के components है homodimer है, but अगर एक alpha एक बीटा constituent है, then you call it as heterodimer. जब इन प्रोटीन्स को लैब में स्टडी किया गया, इनविट्रो स्टडी किया गया, तो ये देखा गया कि ये प्रोटीन्स जो है ये ऑक्टामर भी बना सकते हैं. ऑक्टामर मतलब क्या? 8 सब यूनिट्स जो है वो डीएनए के साथ एसोसिएट हो सकते हैं. दिस रिसेंबल्स जस्ट व्हाट इज प्रेजेंट इन केस ऑफ अ यूकैरियोटिक स्ट्रक्चर, न्यूक्लियोसोम. वहां पर ऑक्टामर होता है हिस्टोन प्रोटीन्स का. तो ये एचयू प्रोटीन्स के क्रिस्टल्स के साथ जब हम लैब में काम करते हैं तो हम देखते हैं कि ये जीनोम के साथ ऑक्टामर बना सकता है. बट अनफॉर्चुनेटली ऐसा कोई भी ऑब्जर्वेशन इन विवो यानी प्रोकैरियोटिक सेल के अंदर नहीं देखा गया. प्रोकैरियोटिक सेल के अंदर हमने डाइमर ही देखे हैं जहां पर 2 सब यूनिट्स ही होते हैं जीनोम के साथ. बट अगर आप लैब में प्रोकैरियोटिक जीनोम और इन प्रोटीन के एसोसिएशन को स्टडी करने की कोशिश करोगे तो तुम देखोगे दैट दे हैव द एबिलिटी टू फॉर्म एन ऑक्टामर. ओके. Now, as I said that we don't have histone proteins. We have histone-like nucleoid structuring protein, which is abbreviated as HNS. That is histone-like nucleoid structuring protein. Now, this protein, which has a role in organization of nucleoid, in organization of the prokaryotic genome, it has specific binding sites. The specific binding site is AT-rich region, where you have lot of adenine thymine repeating regions. That is the site where these can go and bind. Okay, and they are responsible for organizing the nucleoid, and they are also the major component of the nucleoid core. जो हमने core देखा पिछले diagram में. Where did we see that? Yes, this particular core. That core basically has this HNS proteins, HNS family proteins, which are histone-like nucleoid structuring protein. So that forms the basic core, and it binds to AT-rich region where the supercoiling loops are present. Okay. Now, please understand that I told you this earlier also, that we are looking at E. coli as a general scheme for prokaryotic chromosomes, and we are considering that this is how a typical bacterial chromosome is going to look like. But we have to also understand that there can be differences in other bacterial chromosomes and also in the archaeal chromosomes, especially in one group of archaea called as the Crinarchaeota, which has a lot of marine archaebacterial species. Let me tell you that the nucleoid is very similar to that which is found in normal eubacteria. It's very, very similar. But the Urarchaeota group of archaebacteria, they have proteins which are more similar to that of eukaryotic histones, very similar to them. So that also helps you understand the evolutionary link, that okay, Crinarchaeota are the lowest ones, they are resembling the bacteria. Okay, so bacteria may have evolved from Crinarchaeota, but Urarchaeota is showing me proteins which are very similar to that of eukaryotic histones, which means that eukaryotic organisms may have evolved from Urarchaeota. So see, these kind of evidences are also helping you establish the evolutionary linkages. Okay, your phylogenetic studies become easier with these kind of evidences that you are getting. Okay. Now, in some bacterial species, the proteins may not form a dimer. They may form a tetramer. So instead of having 2 subunits, you may find 4 protein subunits which are associated with the genome. And that accommodates around 60 base pairs of the DNA. And the structure looks very similar to that of the eukaryotic nucleosome. Okay, so 60 base pairs of the DNA is being accumulated in that tetramer, and the structure is looking quite similar to nucleosome, because in nucleosome you have octamer, 4 here, and then the genome is winding around it. Okay, so the structure looks very similar to that of the eukaryotic nucleosome in some species. Okay, in other species, the histone-like proteins may form multimers, which are very huge, of various sizes, which can accommodate very long segments of DNA also. But then very less research has been done in that area. Archaebacteria पे ही उनके physical features पर, उनके genomic features पर तुमको बहुत कम research available मिलेगा. Maximum जो तुमको मिलेगा research, that is all available in terms of Eubacteria. Okay, very less details are there which are present about the Archaebacteria and their genome organization or their genome packaging. Now comes very important point. Some bacteria may have linear or multi-parathyroid genome. We will see what this means. अब तक हमने E. coli के genome के बारे में देखा. हमने देखा E. coli का जो genome है वो single, circular, double-stranded DNA molecule. And vast majority of bacteria and archaeal chromosomes also, they show this. But there is a lot of different bacterial species and archaeal species which show linear versions of the DNA, just like in case of eukaryotes. Now, this is the reason why I've been constantly telling you that भले ही E. coli सबसे studied organism है, तुम अगर बाकी के bacterial और archaebacterial species पढ़ोगे तो तुमको vast differences दिखाई देंगे, क्योंकि इनमें circular genome के जगह पर linear version of genome present. First of these which was studied was Borrelia Burgdorferi, which causes a Lyme disease. Lyme disease is very common in America, very less prevalent in India. And this was seen in 1989. And eventually it was found that linear genome is not just in case of Borrelia Burgdorferi, but it's also in Streptomyces coelicolor and also in Agrobacterium tumefaciens, that it is having that linear genome. So linear molecules are going to have free ends, and if it has free ends, then obviously we should be able to distinguish that is this free ends formed because of breakage or it is linear itself from the beginning. क्योंकि जब तुम सर्कुलर मॉलिक्यूल को तोड़ोगे तो तभी भी तो तुमको एंड्स मिलेंगे. तो उनसे डिस्टिंग्विश करने के लिए उन एंड्स पर, उन टर्मिनल स्ट्रक्चर्स पर कुछ तो सीक्वेंसेस होने चाहिए. जैसे ह्यूमन जीनोम में अगर तुम देखोगे तो एंड्स पर टेलोमेरिक रिपीट्स होते हैं. You might have heard of telomeres, right? So similarly, there have to be specific sequences at the linear terminal ends of your prokaryotic linear genome also, which is seen, which is there, which helps you distinguish that, okay, this bacteria has a linear genome and it is not that uska circular genome tha jo do tukdon mein toot gaya hai. So there are specific sequences at the terminal ends. In Borrelia and Agrobacterium, the chromosome ends, you can distinguish them because a covalent linkage is formed between the 5' and 3' ends of polynucleotides in the DNA double helix, which is not normally seen in case of a circular chromosome. So covalent linkage is being formed between the 5' and 3' ends of the double helix. So that tells you that इनका structure ही ऐसा है, ये टूट के नहीं बना है. And in Streptomyces, you will be able to see that linear chromosome ke ends par there are special binding proteins, which are not normally seen. So it's an indication that it's a characteristic feature of these organisms because they have a linear genome and it's not a circular genome. So it's not necessary that if E. coli has a circular genome, all prokaryotic organisms need to have a circular genome. That is not at all true. Okay. Now, a second type of genome is multipartite genome. Now your question would be, what is multipartite genome? So multipartite genome is wherein the DNA is not a single molecule. It's not a single circular molecule or a single linear molecule. No. it is divided into 2 or 3 molecules. Now when I say 2 or 3 molecules, it doesn't mean that वो 2-3 copies है, नहीं, वो 2-3 individual molecules है. जैसे हम वायरसेस में देखते हैं, वायरसेस में जब तुमने इन्फ्लुएंजा वायरस का अगर तुम जीनोम देखोगे तो उसका मल्टीपार्टाइट जीनोम है, अलग अलग सीक्वेंसेस है जीनोम के, and it's not a single molecule, they are split into different molecules. So यानी कि जब बच्चों में, उनके बच्चों में जीनोम दिया जाना चाहिए तो वो 2 या 3 कॉपी, जो 2 या 3 सीक्वेंसेस है अलग अलग, उन तीनों के कॉपीज बच्चों में पास ऑन करने पड़ेंगे, क्योंकि वो डिस्टिंक्ट सीक्वेंसेस हैं, दे आर नॉट कॉपीज. Okay, so when the genome is divided into 2 or more DNA molecules, we call them as multi-parathyroid genome. Okay, now people often confuse this with plasmid, कि प्लाज्मिड जिनमें है उनको हम मल्टीपैराटाइड बोल सकते हैं क्या? नहीं, नहीं. प्लाज्मिड को आइडेंटिफाई करने के लिए स्पेसिफिक जीन सीक्वेंसेस है. Multipartite genome में it's not the plasmid, it is the exact one genome which is present as 2 or more molecules. Plasmid is a very small piece of DNA, but multipartite genome में वो sequences बड़े बड़े होंगे. Plasmid तो बहुत छोटू सा होता है. Okay, mostly it is circular, plasmid is circular, but multipartite genome में they could also be linear molecules. It's not necessary that they have to be circular. Okay, please understand that plasmids can be lost eventually. Organism, a bacteria can lose its plasmid, but a multipartite genome cannot be lost. The multipartite genome contains essential genes which are required for the survival of the organism. If you lose those genes, this organism will not be able to survive. So you have to understand that plasmids and multipartite genome are separate things. Okay, please keep this in mind. Some plasmids, they can integrate into the genome. Not all, very few of them. They have the ability to merge with the genome. But when you talk about other plasmids, mostly they are independent, and if you don't maintain them in a specific condition, those plasmids are lost eventually. Plasmids replication is independent of the chromosomal replication. मतलब जब क्रोमोसोम डिवाइड करेगा तभी ही मैं अपने कॉपीज बनाऊंगा, ऐसा नहीं है. प्लाज्मिड जब चाहे तब अपने कॉपीज बना सकते. प्लाज्मिड का 1 से लेकर हजारों कॉपीज हो सकते हैं एक ही सेल के अंदर. कैन हैव thousands of copies. And when the bacteria divides, the plasmid is also divided amongst the daughter cells. But the process of distribution of plasmids is slightly different. So you have to understand that many a times the daughter cells may not receive plasmid at all. ऐसा भी होता है कि कई बार प्लाज्मिड का कॉपी अगर कम है पेरेंट सेल में तो एक ही को प्लाज्मिड मिलता है, दूसरे को मिलता ही नहीं. बट क्रोमोसोमल डिवीजन के साथ ऐसा कभी नहीं होता. इक्वल डिवीजन होता है दोनों डॉटर सेल्स में, बिकॉज दे कैरी जीन्स व्हिच आर एसेंशियल फॉर सर्वाइवल. वी कैन नॉट एलिमिनेट देम. ओके, plasmids normally carry genes which are not a part of the chromosomal genome. Okay, in most of these cases these genes are non-essential. मतलब plasmid नहीं रहेगा तो organism survive ही नहीं करेगा, ऐसा नहीं होता. क्योंकि plasmid normally special characteristics के लिए कोड करता है. Essential या survival characteristics के लिए कोड करने वाले plasmids बहुत ही कम होते ही नहीं है, बहुत ही rare. Plasmids normally code for characteristics like antibiotic resistance, degradation abilities, and obviously that is going to be only when the bacteria has to survive in such an environmental condition जहां पर उसको ये फेस करना पड़ेगा, तभी ही वो प्लाज्मिड मेंटेन करने का मेहनत लेंगे. वरना क्यों एनर्जी खर्चा करेंगे प्लाज्मिड का कॉपी बनाने में. इसीलिए जब तुम लैब में एंटीबायोटिक रेजिस्टेंट स्ट्रेन्स मेंटेन करते हो जिनका एंटीबायोटिक रेजिस्टेंस प्लाज्मिड एनकोडेड है, अगर तुम मीडिया में एंटीबायोटिक नहीं डालोगे और अगर तुम प्लेन मीडिया पे इस कल्चर को स्ट्रिक करके मेंटेन करने की कोशिश करोगे, that organism is going to lose its plasmid. क्योंकि तुम उसको वो स्ट्रेस दे ही नहीं रहे हो, तो वो स्ट्रेस नहीं है तो वो बोलेगा अरे यार अभी तो मुझे एंटीबायोटिक झेलना नहीं पड़ रहा है, तो मैं प्लाज्मिड क्यों मेंटेन करूं, क्यों रेप्लिकेशन में अपना एटीपी खर्चा करूं, वो प्लाज्मिड दबा देगा. So you have to understand that for a plasmid to be maintained, those conditions have to be provided. Now plasmids can also be passed from one cell to the other by other processes like conjugation. Conjugation एक ऐसा प्रोसेस है जहां पर सेल्स आसपास आ सकते हैं. पिलाई होता है, जिसके पास प्लाज्मिड है they have पिलाई. Okay, that pili acts as a tunnel through which one strand of the plasmid is gifted to the other bacteria. So now जिसके पास प्लाज्मिड नहीं था उसको भी बैक्टीरिया मिल, और उसको भी प्लाज्मिड मिल चुका है from the one which had the plasmid. So वो 2 स्ट्रैंड का प्लाज्मिड होता है, एक स्ट्रैंड तुम बाजू वाले को दे दो, दोनों सेकंड स्ट्रैंड बना सकते हैं, कॉम्प्लीमेंट्री तो होता है स्ट्रैंड. But you have to understand that this is increasing the risk of multiple drug resistance. You all should be knowing about multiple drug resistance. आज ऐसा एक टाइम आ गया है जब बैक्टीरिया एंटीबायोटिक्स को रेजिस्टेंट हो चुके हैं. एसोरियस का एग्जांपल ले लो, स्टेफाइलोकोकस ऑरियस हैज बिकम रेजिस्टेंट टू सो मेनी एंटीबायोटिक्स दैट आर यूज्ड. We are running out of antibiotics to treat Escherichia infection or Mycobacterium tuberculosis infection because the organisms have become resistant to it. So we need to understand that study of plasmids have become very, very significant. Okay. Obviously, plasmids do not just code for antibiotic resistance; they code for other features also. Okay. This is an image which shows you the presence of circular plasmids along with nucleoid in a bacteria. So you can see that there are multiple plasmids in it. There can be multiple number of copies of those plasmids also inside the single cell. So what does plasmid code for? So plasmid can code for resistance, antibiotic resistance. Plasmid can code for fertility, wherein I said that a bacteria which has plasmid will have pili, that tunnel, that channel through which they can share plasmid with another bacteria, can carry out conjugation. So that is fertility, okay, because they are in a way sharing their genetic material. Several plasmids code for toxins to kill competing bacteria. So killer plasmid बोलते हैं उसको. वो टॉक्सिन बनाते हैं ताकि आजू बाजू के कॉम्पिटिंग बैक्टीरिया जो है वो मर जाए. Degradative plasmids, जैसे कि मैंने बोला, some plasmids have genes which encode for degradation process, wherein they they are going to degrade some molecules which are difficult to degrade, like plastic degradation. Many a times the bacteria which degrade plasmid, the ability is plasmid encoded, or rather which degrade plastic, the ability is plasmid encoded. It's not chromosomal encoded. Xenobiotics are those compounds which are difficult to degrade, and the bacteria which have the ability to degrade this, the ability is plasmid encoded. Virulence factors, several virulence factors could be plasmid encoded. Agrobacterium tumefaciens, TI plasmid. The virulence factors are encoded in the plasmid. So you get the plasmid out, the pathogenicity is lost. Okay, fine. So I think that we should stop at this point because I have a meeting today to attend. There's an orientation session that I have to attend. So I will have to stop at this point. But the presentation is there. Please start reading. The reference for this is T.A. Brown. The book is there in the Google Drive that I've shared with you on Wiki. Please start reading the chapter on Prokaryotic Genome. Okay, the points are exactly from your book because I wanted you to know what has to be read and what you can skip for now. Okay, so please use the PPT and the book. T.A. Brown is your reference. Start preparing. Next week, we will continue with prokaryotic chromosome. We will spend 2 more lectures on prokaryotic chromosome before we go to eukaryotic chromosome. Okay? Any questions till this point? Please feel free to ask. If there are any queries, we can discuss them out.
Sir, I had a query.
Sure, please go ahead.
Sir, you said that if we do not use the antibiotics, the plasmids, they do not replicate. If we do not use the specific antibiotics, so how does multiple drug resistance develop if, multiple antibiotics are not being used on one bacteria? If they are in their native conditions, how does it develop?
See, that is the exact point you should know. You know what is funny? आज अगर आप मैनग्रोव्स जाओगे ना, आपको मैनग्रोव्स पता है, राइट? मैनग्रोव इकोसिस्टम पता है ना? मैनग्रोव में जाओ, वहां का पानी लेकर आओ, समंदर में जाओ, वहां का पानी लेकर आओ, आप नदी में जाओ, वहां का पानी लेकर आओ, you will see antibiotic remnants. Do you know this? Because antibiotics are being used at such a large scale. Sewage has antibiotic remnants because इतने carelessly हम antibiotics use कर रहे हैं. पोल्ट्री के लिए, ह्यूमंस के लिए, कैटल के लिए, हर जगह एंटीबायोटिक्स का इस्तेमाल हो रहा है. हम जो एंटीबायोटिक्स खा रहे हैं उसका भी तो मेटाबोलाइट सीवेज में जा रहा है, जो अल्टीमेटली डंप ही हो रहा है ओशन में. तो ये एंटीबायोटिक के रेमिनेंट सब जगह है. तो हमको ऐसे लगता है, बट यू लुक एट द वर्ल्ड पॉपुलेशन एंड लुक एट द अमाउंट ऑफ एंटीबायोटिक्स दैट वी आर कंज्यूमिंग, दिस इज गिवन राइज टू द प्रॉब्लम. पोल्ट्री फार्म में ये धनाधन एंटीबायोटिक यूज होता है बिकॉज उनको पता है कि वहां पे इन्फेक्शन के चांसेस बहुत ज्यादा है पोल्ट्री फार्म में, और अगर एक को इन्फेक्शन हो गया पूरा पोल्ट्री फार्म अफेक्ट हो जाएगा. सो वहां से जो पानी निकलता है which is drained out, that has antibiotic remnants. Waste has antibiotic remnants there. You see in agriculture, to avoid bacterial fungal infection to crops, again antibiotics are being used. So much of antibiotics is actually being utilized that we are not even realizing it. We are looking at just a very small picture. You won't believe that mangrove microorganisms in India are showing maximum antibiotic resistance. Because the kind of stress conditions, all kachra, industrial kachra is going into the ocean. So even antibiotic exact ना हो, उसके जैसा structural analog भी हुआ, उसको भी resistance मिल गया, तो antibiotic को आसानी से resistance मिल जाएगा. Are you understanding this?
Yes, sir.
Anything else?